So this is just, I'm just opened up the Canvas page just to show that where things are. This is your exam review recording. And then if you scroll down, as you can see that all the recordings are underneath the PowerPoint if you need to review. And today's material is the coronavirus SARS-CoV-2 COVID-19 special topic by honors grad students required for all 4 exam. And here is the PowerPoint and the material that I'm recording now that will be underneath this. So this will, I'll just post it just after the class. So that's pretty much about it, that where things are, just for the materials. All right, so I'm just briefly telling the honor students, I'm going to talk to you guys a little bit later. The main problem that what I have is, like many of you guys did a fantastic job, but some of you guys, I can tell that you rushed at the last moment and you did not pick up the proper paper and you put information that is irrelevant to the topic. Remember that for each of the chapter you are given a topic, so you need to stay in there. You do not need to go somewhere else, tell something different. So that's the most important, that's to the point. And a couple of things that when you're in a virology class, make sure that particularly you name the virus properly. It is SARS-CoV-2. When you just write it down, C is capital, O is small, V is capital, and 2. I don't know, everybody else can make a mistake. In your virology class, honor students, you cannot make the mistake. Know the protein, nucleocapsid protein, membrane protein, envelope protein, spike protein. So the name, know the names correctly. So those things are very important. But I'm going to send you an email and talk to a couple of you guys about that. And it's a learning curve. There's no problem. Everybody did a fantastic job. And we'll go from there. You will do much better. So I will just hold on your grading a little bit before I first talk to you. After that, I will post that. So no worries before the exam. We'll do this after the exam, Fridays or Mondays. All right, so first topic that what I had is that how the masks can protect. And you can see that these are done, so this is done by Kirsten, and it's what I wrote is modified. There may be some formatting or something. In some, there's just formatting. Some I had to put material and rewrite those, but that's why I just wrote. But this is original done by Kirsten. So the main point, what you are taking about, and that's one of the things that you are going to do this. Today, the class notes per slide will really help. So that's very important, and I'm going to tell that from each slide, what is your take-home message. So here, what I'm pointing out, let's say your SARS-CoV-2 is approximately 60 to 140 nanometer is a coronavirus size, approximately. So all the coronaviruses, some of them are a little bit smaller because of smaller genomes, about 26, 27 kilobase long. Some of them are a little bit bigger, 34 kilobase long. They're average a little bit better. So why this size varies? The size basically varies. This is just an understanding not needed for your exam. When the virus is budding out, the membrane curves around the nucleocapsid, and sometimes it just puts a little bit extra membrane to make it bigger. Sometimes it picks a tight envelope, it makes it smaller. And that difference gives you the different morphology. So that's basically the overview of the idea. So that is about 0.01 micrometer, 100 nanometer. On the other hand, the virus that we breathe out or that moves out of an infection person, there are 2 things that happen. This is a very interesting idea, and very interesting idea means like, remember that. First is that we produce this respiratory droplets. See that the size is about 5 micrometer, and this is the one which falls within 3 to 6 feet by gravity. When you're so this is the cause for the social distancing. This, if a person is 6 feet, this droplet, which is 5 micrometer, is not going to go to that person. Before that, is going to drop in. Problem is the second. The second is the respiratory droplet nuclei. This is a word that I added, which is commonly known as aerosols. These are less than 2 micron in size, and this can be carried by the air current. And very interestingly, we are understanding that although most of the coronavirus, due to the large size, stay in the droplets and fall in the ground, there are transmission with the aerosol, but the amount is less. That's number one point. Another, just an example, is, say, measles virus is completely carried by these respiratory droplet or aerosol. So basically what can happen that you go to a movie theater, one person is infected and breathing, and the air handler is circulating that, everybody can get infected. Luckily, coronavirus is not that contagious as the measles virus. Otherwise, it could be even like devastating. I mean, like, we think it is, but it could be even worse. So that's the idea. But you can see that here, what is the idea? Mask pore size at 30 micron, 33 micron, 47 micron. So this is how I basically put the idea. Think about this, that you have the front of your Hendrix, you have the couple of doors, you keep all the doors open. And then from here to all the way to the biochemistry department, what you have the other side of the parking lot, genetics biochemistry. You make more walls, and each of them will have the same diameter doors all throughout. And then you put 10,000 people in there and tell them you run straight. How many is going to make it? So that's the concept. Many of them are going to hit the wall. That is the many layer of the mask that is physically blocking it. That's number one. There's a number two as well. That is the most important part, and that will be your take-home message. Mask, right here, let me just, I will just highlight these. All right. So this is the most important part right here. So remember that the mask do not act as a sieve. That is a secondary function. They work because the particles are electrostatically attracted to the fabric fibers in multiple layers removed from the stream of the current. And this is basically, you have these particles, they are all charged. The virus is negatively charged due to the protein, and they are just attracted to these fiber particles. You have cloth, spandex, nylon, they're all charged. And you know that if you have those materials, sometimes you have this electrostatics, you can feel that all the time when it's drier. So over the period of time when it's saturated and the charge can get down and the efficiency of the mask can also get down, but that is the main way that it works. And this is the same principle that your air current handler in your home works. I mean, like your furnace or air conditioning filter. Your dust particles are much smaller. Of course, you can just buy a $2 filter, you can buy a 1200 efficiency, $1,200, $20 filter. But even the $2, $3 filter, which are made of mainly the mesh, block significant amount of this dust that you can see, that you just change. This is the same principle that is in there that works. And if everybody is wearing that, you're just minimizing the chances of the virus ending up on your nose, mouth, from where they can get access to your to the living cells, which does have the ACE2 receptors that we are going to see. So what is the idea? Are you going to just completely contain it? No, you can't. There will be some that will be moving from the side that can pass through, but you are minimizing the number of virus, which is, if you have 10 viruses going onto your nostril on the epithelial cells, your immune system can handle that. But if there are 1,000 or 10,000, there is a threshold limit in there. It is too much for your immune system to handle that. It cannot handle that many of that. So that's basically what we are trying to do. So that's your take-home message, that it does not act as a sieve. It attracts these particles moving through the respiratory droplet or nuclei and remove them. So that's basically reduce the spread of the particles. All right, so after that we are looking at right here. So this is also from Maggie, just gives a very idea that how the respiratory droplets are basically modified in that process in there. that the charged particles are retained. And it is the same thing, stopping the droplets of aerosol by electrostatics. This is the main thing. And limiting the airflow to the outer environment, this reduces the number of virus particles. And if there are two people also wearing the mask, the number of virus particles which can have access to your mucous membrane, which is in your nose and mouth mainly. And that's basically the principle. And the efficiency is 99.8, 97.14, and 95. So really, although of course N95, but you really need an N95? Yeah, if you can and if you're comfortable, that is fine. But still, you can just see that a homemade mask is even 95% efficient in this process. So that's basically what it is. That does not mean that 95% of the virus is contained by the mask. Remember that what we are meaning by the efficiency right here is that the chances of getting past the mask to infect someone, that's basically what it is. So this is the basic principle of that. So again, from the two slides, just remember the principle of that the electrostatics is the main player to contain the virus. It is not the pore size. It is not by the pore size. It's a multiple layer of electrostatic interactions. That's basically what contains the virus. All right, so that's it. After that, we are going to material related to the infectious cycle. And what is the CT value? So when you do an RT-PCR, there is a term that we call, that is called a cycle threshold. That is your CT value, cycle threshold. What it means? That means that you have your sample. Now, you know the basic principle of the PCR. So basically what is happening, that you have a reverse transcriptase is making a piece of DNA from the RNA, and that DNA is being amplified in each cycle. So you have the primers for usually two genes of the, for your coronavirus, you have the specific primers, you have the Taq thermostable polymerases, and you are cycling that. So CT value is that in which amplification cycle you get the signal. That means that you have a positive product above the baseline. Again, it is the cycle at which you are first getting a product that confirms that you have the DNA or the gene present in that sample. So first cycle, amplification means that, so how basically it is happening. Suppose you have one copy of the viral genome in there, one copy, just one copy. And it may take, before you can detect, maybe I'm just saying randomly, 25 cycles before you can detect the product. So CT value becomes 25. Because you have to amplify that product, before that you cannot just detect that. On the other hand, if you have 1,000 copies of the sample in your material, the CT value may be just 11 or 12. In 11, 12 cycle, you can get the product and it's a positive. So the amount of the RNA that is present in your sample can detect that. But there are several things that is also present in there. So what happens, so the first idea of the CT value, that what it is, and what we mean by the detection. So these are done by our real-time PCR machines, where the nucleotides that you put is fluorescently labeled. And you have a laser beam passes through the tube, which is transparent, and you have a detector on the side. As the products are being made, they will fluoresce, and this fluorescence is picked up, and that's your data above that. So usually you will have a kind of a sigmoid curve that you usually see. So right here, what happens, first, just the basic idea, this basic idea, what is a CT value, are generated how early in the RT-PCR cycle positive result is indicated. This is your basic idea. Now, what affects it? Sample collection method, duration of the time between the specimen collection can degrade the viral RNA, increasing the CT values. what happens that you have, so the idea is that if you have a replicating virus in your nostril or in your mouth, you are going to get some viral RNA in there. Now what happens, these RNAs, as they are sitting inside, inside the room temperature in your sample, if it is taking too long to analyze your sample, usually it's done 24 hours, 48 hours within that period of time, but if it's sitting there too long, you have plenty of RNases, and if these RNAs are naked RNAs, they are going to get digested, you are not going to get the result, which is in one sense good. So what happens that when you have the coronavirus infection, after you are even done with the infection, you still have plenty of RNA circulating in your, these RNAs are already fragments of the RNA, or viral RNAs. They are present in your saliva, can be present in your saliva. Nostril may not be that much, but still can be present. These are already the fragmented RNAs and naked, and these are usually digested by the RNAs and will not give you a background or a false positive result. What happens if you have the virus which is actively replicating? Remember that the coronavirus has a nucleocapsid, which is the nucleocapsid is the RNA is covered by the protein, nucleocapsid protein. If it is enwrapped with the protein, the RNAs don't have access to it. So it can sit in the sample for a little while, and then you do the RT-PCR test. At that first step, what you do that you heat the sample to denature that N protein so that the RNA comes out. You put the reverse transcriptase in there. It will make the DNA, complementary DNA copies of that. If you do not remove that N protein or by heating in the first step, you are not going to get the product. So that's the reason that the method works. Basically, that's the reason. But what can happen is that sometimes if a person already recovering or recovered from the disease contains the RNA in the sample, and it is not digested by the RNAs, and quickly done a test and suddenly gets a positive result. At that time, sometimes the CT value can give you a false positive result. The person is not having an active infection, but still can have positive due to these remnants of the RNAs. So these are some of the drawbacks that you can have. And again, this is the whole story. But all you have to do is that so for your exam, these are the things that is needed. The first, just remember what is a CT value and what are the factors that affect the cycle threshold. The main thing is the specimen collection, and definitely underline that RNAs in the sample can degrade the viral RNA, increase the CT values. So RNAs, that's important. And also the type of RT-PCR conducted, primer design, enzyme, and cyclic parameter can also vary. But every day these are being standardized and the method is being better and better. So what happens that when you use the CT values must be correlated first to be used as a viral load indicator, that how much virus you have. A higher viral load can indicate more severe infection is clinically important, but CT values does not represent the viral load. And why it is not? That is what I just basically said, that there could be many different conditions where a person is recovering already. Lots of these fragments of RNA have a fresh sample immediately, RNAs didn't have a chance to degrade that, and it was analyzed, and the person comes up with a positive where the person is really not having much viral load as well. So these are some of the things that you have to consider. But usually if the test is done 1 or 2 times or 2 or 3 times, usually you get an idea that what it is. But these are some of the parameters of the CT value that you have to take care of. So here also, what is the expectation? This is also a very nice slide by Jennifer. This was also the very basic idea of from Rachel. That's perfect. That what definition that what you mean by city. Then here, this is perfect. That overall, this is the idea is that this is the expectation. that if you have the CT value, you do not have to understand that. But the basic idea is that high CT value is a low viral load, negative test result. Low CT value is a high viral load, that is the test result. But does not always directly correlate to the viral load. If a high CT value and the high viral load, if the sample is stored incorrectly, can result in degradation of the viral RNA. So this is the repeat of the same thing, and ineffective collection of the viral sample from the nasopharyngeal swab or the spit test. So there are factors that can be involved in there. So these are some of the things, and see that 16.9 through is not positivity range used for every test which can affect the accuracy of data collected. And the inaccurate data can mislead both public and the healthcare professionals. But what happens that this is the last line, that although we have some limitations in there, but the everyday testing protocols are improving, and now the results are pretty accurate. And always there will be 2, 3, 5% error in the sampling. Some of the tests which has been conducted for 20 years, even your strep throat test, that can even give you a false positive test. So we are pretty good. So just the idea of that, what is a CT value, and just know that the CT value should give you low viral load, high CT value, low CT value, high viral load. This is the correlation. And what are the factors that can affect the CT value, that is important. That's your take-home message. That's it. So definition, correlationship with the viral load, ideally, and what can basically affect that ideal situation, which is many factors that can affect it. So these 3 things, you should be good. Take home. Then we are going into the genomes and genetics. It could get a little intense here. So it's a very busy slide, but just bear with me, and we will go just one thing at a time. So just see that. And then finally, I will tell you that what is the take-home message from there. So first, the SARS-CoV-2 or the coronavirus is a plus-sense single-stranded genome that act as an mRNA ORF 1A, 1B. What is an ORF? ORF is an open reading frame, what the ribosome can read from the start codon to the stop codon. The ribosome reads from a start codon to stop codon, that is an open reading frame. So that's basically can code for a protein or multiple proteins. So what is happening here? Say from 0 to 30,000, this is just a schematic of the full genome length of the coronavirus or the SARS-CoV-2. And so that's the full length. That is your plus strand, the black line, not the green line. The black line is supposedly the genome. In that part, see that right here is an open reading frame 1A, and then another open reading frame is 1B. So you can see that open reading frame here, open reading frame here. The green denotes the mRNA that is produced from the open reading frame. So what happens basically? After the coronavirus gets into the cell, the genome is delivered. It's a complicated process. The ribosome takes up the genome, which is an mRNA. So first, the ribosome translates this as an ORF1A product. It's a protein, which is a polyprotein. And we're going to take that. Many proteins in there, back-to-back joined. They are not separate. Just a single polyprotein. And stops at the end of 1A. There is a stop codon at the end of 1A. But there is a structure at the end of 1A ORF that we are going to learn in the next part. Not that. It is too much for this part. But there is what we call as, just you can keep in your back of your mind, there is a slippery sequence followed by what we call as a pseudo-knot loop. And what happens that sometimes as the ribosome is reading at the stop codon, it should stop. But it encounters that slippery sequence before the stop codon and it slips out of a frame. And at that time, it reads through the stop codon. It cannot stop at the stop codon. If that happens, it makes a B. It ends all the way up to the end of the B. So most of the time, it will stop at 1A. right here, making this copy. That's green part. Sometimes if it slips out of the frame, it will read through all the way to the end and make it as AB. So, or if 1A is translated by the ribosome as 1A and the read through 1AB as the polyproteins. And then what we are looking at, that you take these two mRNAs that is being made, and this is translated into see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Altogether 15 is not clearly given because it's a protease in there. But this is altogether, so 10 proteins are made from here and the additional proteins are made altogether. There are 16 non-structural proteins are made from this 1A and 1AB. Again, 16 non-structural protein, including the RNA-dependent RNA polymerase, which is the hallmark of the RNA virus, is made from that. So how it is made? In this open reading frame, there is a protease in that polyprotein. It's made as a fusion protein, that whole thing, fusion protein. That protease basically cleaves this up. So think about this way. This top blue line, it is made up like a full-length polyprotein like this, or a polyprotein all the way up to this. And then the protease cleaves this one up into these small proteins, and altogether you make 16 non-structural proteins out of that. This is going to do all the modulation of the host cells that we are going to see later, including it will have the RNA-dependent RNA polymerase for the replication and transcription in the second part, as well as it will make up the membrane vesicles where the transcription replication happens. There are some specific proteins which are related with the membrane curvature. But just understand that take-home message, that the polyproteins are cleaved by a viral protease, produce 16 non-structural protein, including RNA-dependent RNA polymerase. This is the first part that is done. Now, as the RNA-dependent RNA polymerase is already produced in the cell, as the infection goes on, the virus set up factories in the cell, in the membrane vesicles, and the RNA-dependent RNA polymerase is now taking again the full-length genome. Remember that. Again, taking the full-length genome, first is make a full-length antigenome. And from that antigenome, it gets back its full-length negative strand, plus strand genome. Or in the transcription, which is shown here, the 3' end, see that this is the 5' end of the RNA, of the genome RNA. This is the 3' end. And all the structural proteins that is present in the virus. So you have the nucleocapsid protein, you have the spike protein, you have the envelope protein, and you have the membrane protein. There are just only 4 protein, structural protein in the virus. All of them are structural protein. They are at the 3' end. And think about the beauty of this process. This 3' end is not translated upon initial infection. First, all these non-structural proteins which needed to make these are produced, and now these green parts are the mRNAs, and see that these what we call as a subgenomic mRNAs. They are much smaller than the genome. These subgenomic mRNAs are manufactured at the later stage of the infection, translated into the spike, envelope, membrane, and the nucleocapsid protein. So this is the process. The first, the non-structural proteins is translated first, and these later. And again, reading, viral subgenomic mRNA synthesis transcription occurs in membrane vesicles called replication transcription complexes after production of the non-structural proteins at the later stage of the infection. And these subgenomic mRNAs are translated into viral structural proteins. So just have an overview of that. How many numbers that we are not going to ask. Just understand that the genome is made into 2 large mRNAs, mRNAs 1A, 1AB, make this polyprotein, make the non-structural protein. At the later stage, it makes the subgenomic mRNA, to make the structural protein. So just those temporal differences, if you just understand that two things happen in two different time points. One, immediately after the genome is delivered, what is being made, and at the later part, after these products of the non-structural protein is made, what is that? That is your take-home message. That's it. All right, we'll ask a very straightforward question from here. All right, so here we are looking at the genomic RNA, and here there are some information in there. So plus genomic RNA is encapsulated with the N protein, that's nucleoprotein, forms the ribonucleoprotein capsid inside the envelope. That's exactly what you are seeing. Genome size is about 2634 kilometer. Again, remember that coronavirus, the Wuhan strain just have approximately a 29,634, or like about 30 kb genome, but that's their average gene. What are some of the features of the genome RNA? You have a 5' cap followed by a leader sequence. This has some implication. It's a sequence which is not made into a protein. That's basically what it is. Then you have some intergenic transcription regulatory sequence and 3' poly A tail. Just know these, but again, I'm not going to ask these questions because these will not make much sense. That what is a leader, what is a TRS, this will not make sense, so that's not needed. So just also know the genome RNA is translation-ready mRNA, but the entire genome is not translated upon the genome delivery, which we saw in the last slide. So this information is important. And part of the viral genome, ORF1A and the ORF1AB from the 5' end is translated into non-structural protein at the initial stage of the infectious cycle. This is again the repeat information from the previous slide that is important. And the viral genome contains the structural coding regions at the 3' end. This is an important information. Remember that. We saw that in the previous slide. See that the non-structural proteins are on the 5. So this is your 5' end. Remember that. This is your 5' end. This is your 3' end. The non-structural proteins are towards the 5' end, where the structural proteins are towards the 3' end. So that's what it is. And these are transcribed after the non-structural proteins are made. And the viral genome is also synthesized at this phase of infection. The genome replication needs a full-length intermediate minus sense antigenome RNA. So right here, let's take a look at what is happening in this process. So right here, after the virus enters into the cell, the genome is delivered. So this is your genome, the green, number 2. See that you see the ribosome, and you see that 1A and the 1B. 1B means that this is 1AB, the read-through. These are the 2 polyproteins that are made and which is cleaved into those smaller proteins, which is not shown. This is the first step. And these proteins set up these double membrane vesicles, which we were talking about in the last part. The mRNA synthesis occurred in vesicle called replication transcription complexes, or RTC. These are those RTCs. So inside, at the later stage, see what happens. At the later stage, the first process is the mRNA synthesis. See that these are making the S, E, M, and N, those 4 proteins. To make that, you have to make an intermediate minor strand, and on that intermediate minor strand, you have to make the plus strand mRNA, which is translated by this, so the S, E, M, and N, and these are translated by the ribosome to make the structural proteins. Also inside the RTs, see that the genome replication happens. In the genome replication, it will not make anything subgenomic. It will make an antigenome from end to end, the full length, and on that minus intermediate, you get the plus strand RNA back full length. So that's the process. It's a little intense, I just know, but this will make the next module a little bit easier as well. So here, see the red arrows denotes the genomic RNA function upon genome delivery. That's where the polyproteins are made, non-structural proteins come out from this. And right here, genome replication, that the genomic RNA synthesis at the later stage of the infection, what is happening, because we're talking about the genomic RNA. So that's basically the whole process. All right, so after that, we are looking at the structure. So this was done by Jerry, and he actually put some more information in there, and I will give you the same thing as an extension for Jerry here. Oh, okay, did a good job. So your next part of the protein-protein interaction, that will go into the module 2. So you can use the same literature to do that. So, here, so I had to, I cut down a material which is just pertaining to this material. So what is happening right here, you can see that it is a plus strand RNA, we already know, with a helical nucleocapsid, which is inside right here. So that's your important, absolutely know that. The N protein, the nucleocapsid protein, is multivalent, interacts with multiple protein and RNA. What we are going to see that here you are seeing the nucleocapsid protein is interacting with this M, or the membrane protein, which is on the envelope. At the later stage, what I was talking to him, you will see that the N protein will interact with many other proteins in the replication transcription complex. But at this point, that's not needed. So what it does, that the N protein packs the RNA genome into loops and tether the nucleocapsid with the M protein. So the interaction, that word is not needed, sorry. All right. That will make sense. So nucleocapsid with the M protein. So it keeps organized the nucleocapsid. And the N-terminal domain, or NTD, these are mainly beta sheets with a positively charged amino acid for the RNA binding. So here is your, here is your important in-between line question right here. So the N-terminal domain, mainly beta sheet with a positively charged amino acid for RNA binding. Your take-home message from here. Why it is so? Your RNA with the phosphate backbone is negatively charged. It is attracted to the positively charged amino acid. So this is an important part, that so the question could be like this, that why the RNA binds, where the RNA binds and why it binds. So just remember that it is due to the positively charged amino acid in the N-terminal domain. That's your very important question that you can take here. And the C-terminal domain is the dimerization domain to form the structure of the genomic wraps around. This is mainly alpha helix. So you can easily see right here, this is your N-terminal domain. This is contains positively charged amino acid, binds the RNA. This is the C-terminal domain. And see that this is one N protein, N-terminal, C-terminal. This is the next one. And the C-terminal domains are dimerizes. And this makes a structure like this. And if you have taken other molecular biology or something like that, it is not like that. Don't quote on me. But nucleosomes actually pack the DNA just like the histones, nucleosomes are packed like that. So you can see that it is very efficiently packs that genome into the nucleocapsid. So that's basically the nucleocapsid structure. And it also, the important part that you can see, a part of the membrane protein also tethers the N protein, so the genome is properly organized. It is a large genome. You have to properly organize it, otherwise it's not going to fit the whole thing. So that's basically your nucleocapsid structure. So after that, a lot of information here, the SARS-CoV-2 envelope, and you have 3 proteins in there. So the first, what is the envelope? The envelope is derived from the cellular endomembrane, primarily from the endoplasmic reticulum Golgi intermediate complexes. So these are, some of the viruses get their envelope from the plasma membrane, from nuclear membrane. Here you know that they are from the ERGIC, or the endoplasmic reticulum, or Golgi intermediate complexes. There is a picture which you can probably correlate. See right here, this is your ERGIC. See right here, this is the naked nucleocapsid. And here it is acquiring the envelope right here, is acquiring the envelope from the ERGIC. So you can correlate that. So what are the different proteins? So just know the basic thing. The membrane protein is a central organization required for assembly and membrane curvature and the budding. So mostly you can see that These does have these transmembrane domains, and they basically bend the membrane so that to form the envelope. This is one of the main functions for them. And the cytoplasmic domain tether the nucleocapsid protein to organize with that. This is the structure. And again, so you may ask that what we are asking for the structures. No, this is just for your pictorial view that what it is. And you can see that the ones S4, A2, D3, T175, these are the ones which are the space filling. Those are the one which are the mutation hotspots. You see those in different strains of the coronaviruses. That's what it is. But here we are not talking about that at all. We will talk it somewhere else. Next is an envelope protein, which is just 75 amino acid. So it's a very small protein. But it's a pentameric viroporin-like protein. So you know that what purine, an aquaporin is an aqueous channel. So you can see that the side structure of the E protein is like that. But if you look at from the top, you have 1, 2, 3, 4, 5. You have a pentamer of each of these proteins. So you can see that it makes a kind of a channel through the membrane. And this is essential for membrane curvature, assembly, and it interacts with two other proteins called NSP2 and NSP3. We're going to talk about that somewhere else to set up these membrane vesicles. That is very important as well. And this also induces cytotoxicity. So you can just easily imagine that what is going to do in the cytotoxicity. Some of these viroporins are going to be transported onto the plasma membrane. This is not needed. Onto the plasma membrane. And these are basically open channels, make the plasma membrane porous, kill the cell. Virus exits the cell. Once it's done, leave some of this viroporin, which goes onto the plasma membrane and make holes on the plasma membrane, making the cell dead. So that's basically the how it can kill the cell. So amazing process. Just not only the envelope structure for the structural integrity, but also that's how it kills the cell after budding. Then the most famous one, the spike protein, 1273 amino acids. It's a large protein, 122 or 200 kilodalton. It's homotrimeric class 1 fusion protein. So this is one of the important things that definitely remember that, because we already know the class 1 fusion protein. We have seen that for the hemagglutinin in detail for the influenza virus. We have already seen that. So that's why that is important. And this is the protein which binds to the angiotensin converting enzyme 2 receptor implicated in the fusion entry. In this slide, just only remember that it contains two domains. S1, large receptor binding domain, and it is the receptor binding domain, we will refer that as RBD, receptor binding domain. This is the receptor binding domain is the top. This is the receptor binding domain. And then let me change the color. And this is the stalk of the spike. This is the stalk of the spike. So that's basically the two differences in there. So definitely remember that the S2 is that here you are looking at the S2 from the top view. If you are looking from the top, you can see that you have a monomer here, monomer here, monomer here. It's a trimer, and that binds to the ACE2 receptor. So this is the overview of the whole structure, and we are going to see that what these means. This is your N-terminal domain, receptor binding domain, and there are some cleavage sites. The fusion peptide is right there. That basically inserted in the membrane for the fusion, and then so there are some heptide repeats. We are going to see that what they are in a second. So just basic idea, again, membrane protein, abundant central organization required for virus assembly, membrane curvature, budding, and tethered nucleotides, the nucleocapsid, envelope protein, pentameric viroporin, induce cytotoxicity, homotrimeric class 1 fusion protein, mainly implicated for the receptor binding. That's your take-home message. That's it. All right, so this is a slide that I just added just to have an overview of the subunit S1 and S2. And in the three-dimensional structure where you have that, so right here, the bottom portion where you see the head groups, these are the phospholipid bilayer. just to show that where they are. So this portion is not needed for your exam, just to show you that where the things are. HR1 and HR2 are called the heptide repeats. These are 7 amino acid long repeat structures, and the fusion protein is tucked in right there. Now, this is an understanding that understand very carefully. There is a couple of protease cleavage site in between the S1 and S2. S1 first binds to the ACE2 receptor, and then either in the on the plasma membrane, or if it is endocytosed in the endosome, there is a cleavage in between S1 and S2, and the S1 moves away. As S1 moves away, this fusion protein is extended and inserted into the membrane, which causes the membrane fusion. That's your first take-home message. So let's look at the couple structure. So this is your N-terminal in here. This is your receptor binding domain of the S1, which is right here. And right here, in between the S1 and the S2 subunit, S2 is the stalk, you have this specific site, which is the unique mark. And this R is unique among the amino acids. And this is what is called a polybasic site. You can see that these are the basic amino acids, and these are the arginines, and these are the polybasic sites. And SARS-CoV-2 acquired one extra polybasic site, and that's probably one of the reasons that suddenly it just got more infectious, in the sense chances of proteases cleaving it out for entry has increased due to that site. That's basically the hypothesis. So let's take a look at what exactly happens. These are just some other ideas, space filling model, and you can see that the purple colors are the conserved residues, and the variable residues are mainly at the top. So this is where the mutations are happening, and all these variants are happening. See that the stalk is not changed much. This is not needed for the interaction with the host. What is being changed is basically these residues which interact with the receptor. And this is an intelligent selection of a molecule which is completely passive. It is just by chance. And that's the unique part of it. And again, nothing needed, just for your understanding of the class 1 fusion protein, where the things are. All right. So here also another detail, and again, it may feel a little bit overwhelming, but what we are pointing in here is that what we mean by acquired an extra protease site. So here you can see that in between S2 and S1, you have a site which is a protease site, and this is an S2 prime site, and this is present in all coronaviruses. So one of these sites, so remember that, one of this site and this site, these two sites are present, this one and this one, present in all coronaviruses. On the other hand, what happens that right here, you can see that right here is an arginine, this is a polybasic site. This is a furin-like protease cleavage site. This is an extra one which is acquired in novel in the SARS-CoV-2. So you have added this as a separate site. So you have already this and this in all of them, all coronaviruses, you have an extra site in there. So that basically, when you cleave by the protease, that domain, the S1 domain, right here is your fusion peptide. This is an internal fusion peptide, IFP, and this is exposed. So basically what is happening in here, adding an extra protease site just increases the chances of cell membrane protease to find two of them to remove them. Instead of finding two sites, now there is an extra, and that subtle thing is make the virus more infectious. That's the basic idea to understand that. So just remember that for the SARS-CoV-2, it acquired one extra protease site. That's your take-home message from from this. So what is happening then, the role of the spike protein. So the next part that what we are going into is the attachment entry, of course. So again, some repeating information containing S1, S2, two potential protease sites, cleavage sites between them. S1 contains receptor binding domain found in the open and closed conformation as well. The open conformation of the RBD binds to the ACE2 receptors. Once they are bound to the ACE2 receptor on the plasma membrane, there are many different proteases. One of them which is implicated is a human transmembrane protease serine 2. It's called TMPRSS2. And also there are many other furin-like proteins, and these are called proprotein convertases. They are inside the cell and outside the cell. Does many different types of function. And these proteins basically cleave that in between the S1, S2, that what you see. And after that part is cleaved off, a conformational change in the S2 subunit allows the fish 1 peptide to be inserted into the membrane, triggers the 2 heptapide repeat sequence to interact, and pull the viral membrane closer to the membrane to fuse it together. That's just like a perfect definition that how exactly this is happening. So let's take a look. So that's basically the repeat information. So just understand that the S1 and S2 separated. Again, repeat information. So we will ask that, and the fusion peptide is exposed, inserted into the membrane, and that's the mechanism. Just the basic mechanism, that's good enough. On the other hand, you can see that right here, if you just correlate that, again, I'm not going to ask that, if these are the heparin sulfates. So these heparin sulfates, if you just go back to your attachment entry, you will see that sialic acid and the heparin sulfates are not receptors. They are enhancer molecules and they are charged. And initially, just by the electrostatics, the virus actually binds onto those. And then, as they bind, a lot of them, they're trying to find the ACE2 receptor. Once it finds, then it lodges in. And remember that as the ACE2 receptor is bound by the spike protein, immediately there are two things can happen. Once, the proteases can chew them up for the entry at the plasma membrane. But there is another surprise that is left. So that is now, we'll unfold that. So let's take a look at what is happening here. So let's go through this. The S1 ACE2 receptor 2 subunit mediate the membrane fusion. Cleavage by the host protease activates ACE2. Furin cleaves the C1, C2 sites or the pre-cleavage. This is C1, C2 is basically the S1, S2 prime sites, basically. The converted sites, so don't confuse with that. TMPRSS further cleaves the C2. And the cathepsin B and L are also involved. So remember that this is just what we call as, so you already S1, S2, and the S2 prime. So this is what basically being told in here. Instead of S, we use the C1, C2, and that's what it is. And what we are going to see that cathepsin B and L are also involved in the endosomal entry pathway. We are going to see that in a second, but here, this is what is needed. Just remember that at the plasma membrane, you have the TMPRSS and the furin. These are the two proteases that is implicated for removal of the S1 subunit. So what is happening? ACE2 is binding right here, and the furin or TMPRSS2, either one of them, they can cut that S1 out, and this S2 exposes the peptide. The S2 is right here. It is converted to that, and that will insert into the membrane. And we are going to see that in a second, that how it happens. The animation will make it much, much clearer. So first we are looking at, so these are the proteases that are involved. So just remember that, again repeating. The TMPRSS and the furin that acts on the plasma membrane, and also cathepsin B and L are implicated in the endosomal entry pathways. So here what happens, we are looking at the plasma membrane at the neutral pH. So here, there's the same thing. We are going to look at the animation right here. You have the virus. This is S1, S2 together. It locks onto S2, and tempress right here is being shown. That could be a furin. Cuts this piece out, and the S2 stalk unfolds and inserts onto the membrane and pulls these two down. We're going to see an animation. So that's, again, another thing. And camostat is basically a drug that can stop this process. It's an inhibitor of this protease tempress 2, but that does not stop the viral infection. So the scientists found that there are many other ways that the virus can enter the cell. So we are going to see that. Again, this is the repeat information. What are the proteases? We are repeating that, so that's why we're repeating the information. So you'll see that there is lots of redundant material, so that's why although there are a couple of slides, but many of the slides have the repeating information. And the proteolytic cleavage exposes the hydrophobic amino acid and then fold themselves onto the viral membrane fusion. So then Janet Iwasa's lab actually did a fantastic job. And look at this animation. It's a little fast, but I actually downloaded the movie and put some pointers in there by editing those in there. So that may help a little bit because it was a kind of too fast. So what we are looking at the envelope, and you can see that the M and the E protein, envelope proteins and the E proteins right here. And this is the famous spike. And the gray part that you are seeing on the spike, those are the glycosylations. And these are built after the molecular models. These are just not cartoons as such. So let's take a look at this. Nucleocapsid, if you just cut it open, all are molecular models. Now tempress comes in after it's being locked onto the ACE2. Right there, takes out the 3 trimers of the S1, and it is left with the S2, which is the stalk. The top head is removed. Now the fusion peptide falls right there onto the membrane. And the heptide repeats, which are in the middle, is going to interact right here. The heptide repeats, and that's your membrane fusion. I mean, like when you have the structural biology, this is the kind of information that you can get. So that's basically what it is. I think that it's probably one of the coolest understanding of the movie, that if you just see this movie, you know that exactly what happened. All these talks, even if it is confusing, just watch that and correspond to that material. The next thing that what we are looking at, that also along with that, we have the virus has the ability to enter the cell through the endocytic vesicles. So what is happening in there? So just remember this, that these ACE2 receptors, they are on the plasma membrane, and there are lots of these clathrin-coated pits that is constantly being formed on the surface of the cell. So clathrins are basically clamping type of molecule. They just pinch the plasma membrane, make a vesicle. Many of the time, it just brings in the receptors. If it's not, it's empty, it just recycles the membrane back. And this is constantly happening. All these cells are making these clathrin-coated pills, being pinched off and recycled back. If some of these ACE2 are found on a clathrin-coated pit, as soon as the virus binds to the receptor with the S1, that trimer, before the tempress or the furin can basically cleave that, before that the whole thing is endocytosed. Remember that. So this is your clathrin-coated pit. The spike protein directly binds with the S1, just in that. But it doesn't have enough time for the plasma membrane protease to act on that and remove that. If it can, it will enter through the plasma membrane. But if it is endocytosed immediately, it just ends up into the endosome. And in the endosome, what's going to happen, that as you know, in the late endosome, it's going to pump the H plus or the protons, and it will fuse with the lysosomes, and it will release this cathepsin L and B. These are the proteases. These can also cleave the S2, S1, causing the fusion peptide to open. And the same way, remember that the plasma membrane, you have phospholipid bilayer here, and the endosome also have the phospholipid bilayer, and the mechanism is the same. Just a different protease acting on them in a different pH. At neutral pH, it is either TMPRSS2 or furin-like proteases. In the endosome, if the acidic condition, it is cathepsin B or L. But the mechanism of fusion is the same. So this pathway can be stopped by hydroxychloroquine. That is implicated, and again with the controversy. But most of them entering through the plasma membrane. So if you're just taking the hydroxychloroquine, you can stop the endocytic pathway, but you cannot stop them from the plasma membrane. So we saw, and there is another drug that we saw there, which is Chemostat. So it's like, okay, all right, so you take Chemostat and hydroxychloroquine. Now we have a magic drug, a double drug. So we should not get anything. Unfortunately, that doesn't happen because of the last line right here. Caveolin-dependent pH neutral as well as clathrin caveolin acidic independent pathways being implicated as well. So there is always a way a virus will find to get into the cell. So even with these two drugs, cannot stop those pathways where those works. I mean, like antivirals are still being developed. It will take more time than the vaccine, but that's what it is. So that's pretty much, I guess, is the overview of that. So 908, and what we will do that, so that's the overview. So we have about 10 minutes. If you just, anyone have a little bit, it's quite a bit of information, but I said that from each of the slide, what are the, say from here, acidic, from this slide, you're again, your take-home message will be, it's acidic pH, cathepsin L or B cleaves the same thing. That's basically what it is. And we are not going into the hydroxychloroquine or the Chemostat. We are not going to ask. We have a chapter called antivirals. We're going to ask. So we are not going to ask that. This is just an overview of that. So now what you can do that as we do not have much material written in there, just the PowerPoint slide, you can ask for a particular slide. I can just repeat or explain, or if you think that from that slide you are not sure that from a particular slide what is the take-home message, I can repeat that. So please do ask. Any one of them? So I'm going to go one at a time. So even if you're, so we are good with the mask. Good? So here we're looking at just multiple layers of electrostatic interactions. That's what it is. Size doesn't matter. So that's what it is. That's your take-home message. So here, CT values, remember that the CT values, low viral load, low CT value, high viral load, and generated by how early in the RT-PCR positive result is found, and what are the factors that can affect the CT values. So specimen collection to analysis time, RNAs in the samples. So your RNAs is the most important one, which can degrade. RNAs is everywhere. It's kind of really super funny. On your palm, on your skin, I mean, like there's a tons of RNAs. Anybody works in a lab with an RNA? Anyone? So just ask that they cannot just use even autoclave water. They have to add a chemical called DEPC, diethyl pyrocarbonate. Then you autoclave the water. That can kill. So it's, the RNAs are just rich with the beta strands. Autoclave doesn't even deactivate them. So they have to treat the water with DPC overnight and then autoclave it. So really difficult. Like difficult means like you have to be like super careful while working with that. All right, so that's we already saw. Here, it's a complicated slide. If you just ask anything specific, I will be more than happy to ask. Just remember that the full length up to 30,000, that is the full length of the genome. Only two portions overlapping is made into the non-structural protein in the first part of the infectious cycle, or translated in the first part as the virus goes in. Once these non-structural proteins are made, then these set up these viral factories in those RTCs, or the replication transcription complexes. They take the genome RNA as the template, and from there they make the mRNAs, because this is mRNAs. That's why we are saying that they are making the mRNAs, which code for these proteins. That's basically what it is. And this is what we call as a subgenomic mRNAs, much smaller than the genome size. And also, in the same way, this is what we talked about in the last one, subgenomic mRNAs. And also it makes the full length of the antigenome, which is the minus sense in olive, makes the plus sense full genome. This will be packaged inside the virus. And the mRNAs are translated into the proteins, which are the A, C, E, M, and N. And that's what it is. It's interesting that this is just extra information. Some of the coronavirus actually do have another protein on the membrane, which is called hemagglutinin esterase, HE. That is not there. It's not present in the SARS-CoV-2. All right. So any questions from these two slides, because these are a little bit busy? Everybody good? Okay. So I think that just remember that why the N-terminal domain contain the positively charged amino acid for the RNA binding, that's important. And the C-terminal domain is a dimerization domain. And the nucleocapsid is a multivalent. It binds RNA as well as protein-protein interaction, and it's interact with the M protein, which tether the nucleocapsid to the membrane. Just the overview of that, the membrane protein, central organization for viral assembly, membrane curvature budding. Envelope is a viroporin, that underline that word, viroporin-like protein. You can see that the channel is in the middle. And it's required for assembly budding, also induce the cytotoxicity. And the spike protein, you already know that receptor binding and the stalk. That's all good. These are the repeated information. You have already seen that. So just to remember that the furin-like, this is the extra site that is acquired in the SARS-CoV-2. So you already had two sites, but this is the one which is the extra site that it acquired. Conformational change, that how it happens, that S1 is removed, exposes the fusion peptide, falls onto the membrane, and if you just see the movie, it's very close. So the only thing that what you have to remember, that furin-like proteases, which is a class of protease. Furin is a protease. Those classes are called proprotein convertases, and furin is one of them. And temperase, these are on the plasma membrane. They are not, they act in neutral pH. Furin is also in the cytoplasm, does different functions. Where the cathepsin B and L acts as an acidic pH, and they are in the endosome. So for the endosomal entry. So we have seen the whole process. Definitely look at the video, just a little bit idea. And right here in the acidic pH. So just remember that, oops. I thought you dropped the laptop. It just skipped my heart. So just remember that when you have this is the conceptual question, that for the when you have the endosome being formed, cathepsin B and L cleave the S1. It binds to the receptor, but in the plasma membrane, the S1 is not removed. The whole thing goes into the endosome. So remember that will be your trick question right there. That in the endosomal pathway, the S1 is not removed as it happens in the plasma membrane entry. So everybody good with that? So that will be your one trick question right there. The rest will be all straightforward as said. But if I have said the trick question, don't miss it. All right, good luck. And again, you will see that on your exam, 50 questions, 75 minutes, 2 points each. And you're going to see it will open at 6 o'clock, open until 11:59 p.m. You will be able to see all 50 questions in one page. No one question, all those kind of things. So you do not have to flag. You can just go anywhere and take your time. That's it. But make sure that you have the latest version of the Respondas and just do that silly quiz just to check that it happens. Sometimes it will not pass through if you have an older version. Download it from the CCID website. And if you have a difficulty, I will give my Google Voice number on the top. Just call me anytime. All right, good luck. All the best.